There we are. Oh my God. Look at my hair. Hello. Hello, people. Oops. Keegan Dupree. Bing. We're recording. Oh, okay. Here we are. Okay, people. Looks like I can mute or unmute you if I want. That's fun. Rachel Siller. Welcome, Rachel. I'm waiting for Christina to come because she had some questions I want to deal with. A question on the question forum. Okay, anybody have any questions before? Any questions? Yes, I have not had a haircut in over a month. Getting long. I'm still thinking about that comb over, bringing it right down to here and like square across, like the old days. No questions. You guys are all muted. Are you trying to ask me something? Why doesn't somebody say something? Say something. Christina said she's not home right now. Oh, so Christina's not coming. Okay. Hello, iPhone. Welcome. Okay, does anybody have any questions? Or should I do my PowerPoint or what do you want? I'm at your beck and call right now. Can you guys hear me? Can you respond? Okay, Sydney. Can you guys hear me? Can anybody hear me? Okay. All right. So yes, one more exam and then that's it. We're calling it a day. That'll be 4 exams total that will average up. Oh, Yvonne. Sorry, Yvonne, you're welcome. Okay, let me pull my PowerPoint up. If I can remember how. Share screen. Okay, so there's this. You guys okay with this from last time? If so, we move on. Okay, so one more thing to finish up about Hox genes. The very first thing I talk about in this set of notes is the regulation of the Hox gene. So in this slide 22, I was talking about how the Hox genes get expressed and then laid down into the somites, and that patterns the somites because you've got this wave of Hox gene expression going in this direction as the node is going in this direction, and you're laying down your pattern. Yeehaw. Okay, so slide 23 then, excuse me, we're talking about kind of how that happens. We start talking about how that happens. So if you remember, we've got a lot of Wnt signaling going on at this point. The node is releasing Wnt. I believe all the cells that are in that primitive streak are releasing FGF and Wnt. So if you look at the gene for, question? So if you look at the gene for a Hox cluster, at the 3' end of that gene, there's a, okay, hang on. Dog needs to go out. I'll be right back. Stay out. I'm back. Okay. Share remote control. All right, well, I'm just going to go. Well, hang on a second. When I got up there, I'm afraid someone might have tried to get in, and my my settings aren't right, so I have to allow you to come in. So let me just check to make sure. Okay, I'm going to keep this over here. All right, so how are these Hox genes regulated? So you've got this 3' area here, which is an enhancer. Hopefully you've had genetics and you know what an enhancer is, but basically proteins bind to it, and then that brings this part of the DNA over to the promoter of the gene you're activating, and that helps it to activate. So you need that enhancer to be bound with specific proteins to turn the genes on. So Wnt signals activate transcription factors, and those transcription factors bind here in this 3' end, and then that activates gene 1, HOXA1 here. So at the same time it's activating HOXA1 from a different chromosome somewhere else, it's activating CDX2. Remember CDX2? That was early on in mouse development. It was involved in trophoblast versus hypoblast differentiation. So here we see it showing up again. It's also a transcription factor. So when you activate the first genes, you also activate CDX2. And if we skip down to the next slide, CDX2 then activates the next genes in the row. So they're activating, so the forward genes are activating the later genes. And then when you get all the way to the last gene, HOXA13, it starts to, it will inhibit the previous genes. So they're activating from the first to the last, because the first one that gets activated is because of Wnt signaling, and then later ones get activated because of the earlier ones until you get to the last one shuts off the rest of them. So that's why they're coming out in that order. Okay, is that clear? That's all that first thing says. Okay. Okay. Moving on. So what are we going to talk about today? So this Hox gene expression is giving us an anterior posterior patterning down the body plan. Now we're going to look at each individual somite and how is a somite patterned between the dorsal and ventral side, the anterior posterior side, and lateral medial patterning. So this is what's going to, so we're getting further, we're not just generic front to back now, we're getting into actual structures now, starting to make tissues. So first thing to look at is with the anterior posterior development within a somite. So I'm going to draw a little bit here. So here are the somites, kind of cuboid looking. Okay, maybe this is the anterior part of the animal. This is the posterior part of the animal. So within this, between the anterior side and the posterior side of the somite, that's what we're looking at now. So what happens is back here, you've got notch being expressed. Oh, shit. I got to pause because I have to take this call. Hello? It is. I'm in a Zoom meeting right now. Can I call you back? Do you have a direct line or name? Mm-hmm. I'm sorry. Say it again. Konn, Konn. Okie doke. Thank you, Doc. Sorry about that. I'm having a problem with my. Boy, this is the worst Zoom you guys have ever seen. Okay, so here we are. So at the posterior side of the somite, you've got notch being expressed, notch and delta. So there's like, I think delta over here and notch over there. If you remember, delta is the ligand for the notch receptor. So you've got delta on one side and notch on the other, and that's what makes them separate. So you get two, that's what makes them separate like between somites. Up here at the other side of it, you've got this MSP. So one side of the somite is expressing notch, the other side is MSP. And you can kind of see that here in the figure in that in the top one, they're showing you just the delta expression, which correlates with the notch expression. And in the bottom one, they're showing you MSP. So you can see this kind of striped where you've got MSP at the anterior side and nothing here, but that's where your notch and delta are. Okay, so anterior posterior patterning within a somite is because of notch delta versus MSP. And then probably more interesting now is when you start looking at dorsal ventral. So dorsal is the back, ventral the front. Here's the back. Here's your neural tube. Let me bring this up a tad. So here's your neural tube is already closed up, nodal cord underneath it on the ventral side of it. And then here's the somite on this side of the neural tube. So now this somite is going to give us three different tissues really. One tissue is down here in blue, it's the sclerotome, and that will become spinal cord. And then the other part, the yellow part, is dermomyotome. And you can tell from the name itself that you've got two different tissues there. Dermo meaning skin, myo meaning muscle. So you've got both skin and muscle forming here in the top part of this somite. And what that differentiates into depends on signaling that's happening at the time. So we come down to the next slide. Oh, and by the way, this is a really good animation. The guy's a little boring, but he does explain things well. Okay, so here's your dermomyotome. Now that's going to differentiate into muscle on the sides and skin on the top. And when I say skin, it's the dermis, which is the layer of skin underneath the epidermis. So epidermis is just squamous cells, flat squamous cells protecting. The dermis has got blood vessels and all kinds of smooth muscle and things like that. So this is going to be the dermis. And so on the side, we've got cells migrating out to become muscle that's going to be more lateral. And on this side, you've got muscle cells migrating to stay near the spine to become the back muscles. So let me pull that up real quick. Okay, so I'm down here, dorsal ventral patterning. So you've got your dermomyotome, and this is dermomyotome because it's expressing PAX3, transcription factor. That makes it dermomyotome. And your myotome then will express MyoD. MyoD is a classic transcription factor for muscle differentiation. So we're going to get muscle with this. And the ones that are medial, close to the neural tube, are going to be axial and back muscles. So everything going up the middle. And then the lateral ones will be abdominal muscles and limbs, when the limbs come out, where the limbs come out. Dermomyotome then is on the dorsal side, which makes sense because your skin's on that dorsal side, and that becomes the dermis underneath the skin, and the ectoderm there becomes that epidermis. Sclerotome then is on the ventral part of the somite. That's the blue cells. Sclerotome then is expressing PAX1, which is why it's sclerotome, and it will form the vertebrae and the ribs. So this is going to be bone, and bone, both like developmentally and also evolutionarily, starts as cartilage, and cartilage chondrocytes become osteoblasts, and that becomes bone. So it starts out as cartilage and eventually becomes bone. And this dorsal ventral patterning is because of some really cool signaling interventions here. If we go to the next slide. Okay, so we're on 29. So you've got, shoot. So you've got signaling coming from different directions here. From up on the dorsal side, you've got wince coming in, and that wince coming from the ectoderm and also from the dorsal part of the neurotube. So you've got a wince gradient from the top to the bottom, high, low. Then there's a gradient going in the opposite direction, and this is a new signaling pathway that we haven't talked about yet, and that's sonic hedgehog. And so sonic hedgehog's coming from the bottom with a gradient to the top. And then we've got BMP4, which is coming from that lateral mesoderm area and is coming in. So now you've got lateral to medial gradient of BMP4. So those three gradients are what's creating these three different tissues here. Sonic hedgehog, by the way, I don't know if I've ever told you the story. I always tell the story because I think it's hilarious. I was working in this lab at Harvard Med School when I knew the guy who named this Sonic Hedgehog because his son liked that video game. It was another hedgehog analog, and so he called it Sonic. And actually, I was reading up on it recently, and a lot of doctors got really mad about it because it sounds really silly, Sonic Hedgehog, and it's involved in a lot of cancer. So it's hard to explain to a person that they're dying because of Sonic Hedgehog. It's embarrassing. Okay, so does this make sense, I hope? Your Sonic Hedgehog is turning the cells in the bottom of the somite into chondrocytes, and they're moving to become the vertebrae and the ribs. The winch is causing the stuff on the top to become dermis. And then you've got this BMP gradient over here that's determining medial versus lateral of that mesoderm, the muscle there, okay? So you've got your lateral mesoderm because it's high BMP4 and axial, axial and back muscles over here and low BMP4, and then limb and abdominal over here where it's high BMP4. Okay, does that make sense? I'm not getting much feedback today. Can I get a yes? Give me a yes or a no. I'll go slower. I'll go, look, you guys can interact with me over here. See this? Start beeping on that. Are you guys watching? Are you guys like downloading the soccer tournament or something here? Oh, there can't be any soccer tournaments. Can you guys even hear me? Am I talking to myself? What's going on?
You can unmute us.
About the car.
One plan of action is to dictate his right. Is not
You
one
said you saw check him out?
but his personality.
Yeah, I just unmuted everybody. I think I hear someone's television.
With a gazer in disguise.
What's wrong with that?
We can hear you.
You can hear me, good. So that's all I have for today. Well, one more thing, but I just want to make sure everyone understands that. Are we good?
Yeah.
Yeah, okay. So the only other thing to talk
Yeah,
about,
bro. What's wrong? Yeah, if it was a double, not that.
oh, it's Justin.
They wild.
Justin,
Hold on, bro. Hey.
hey, you're unmuted there. So just
Oh,
so,
sorry about that.
that's okay. Just so you, oh, I unmuted you on purpose. Rachel's unmuted, but she's quiet, so you wouldn't know. Okay. All right, so last slide then is Sonic Hedgehog pathway. This is a weird one. Sonic Hedgehog, basically, the idea is you've got, again, some of these silly names as participants in the signaling pathway. You've got your primary thing that we're regulating is this protein over here called GLE. So GLE is a transcription factor. And did someone just come in? What was that beep? I don't know. So GLE is a transcription factor, and it can either be a repressor or an activator of gene transcription, depending on how it's regulated. Oh, chat. Where's my chat? Alt H. Is someone chatting me or what? Ugh, not you. All right. Oh, there's my chat. Is this one of the pathways that was in the Cell Bio games? I think so. I think it was in, not Cell Bio, but in Bio of Cancer, which this year I threw into Cell Bio as well. I think this was up there. But anyway, the whole idea is we're going to be regulating GLE. And GLE is regulated by SmoothEnd. SmoothEnd is in a vesicle in the below the membrane, and GLE is up in what's called a primary cilium. It's a cilium, but it isn't used for motion. It's used for signal transduction, kind of a newish thing. So SmoothEnd is in this vesicle below the membrane, and GLE is up in this primary cilium. Now, if SmoothEnd can't get to GLE, then GLE comes out, gets cleaved, and gets turned into a repressor. So that's what's going on here. SmoothEnd is blocked by patched So glee comes out, it gets cleaved, it binds with some other factors and becomes a gene repressor. When hedgehog is around, hedgehog binds to patch, patch being the receptor. Patch no longer blocks smoothen. Smoothen then via, if you're in cell biology, beta arrestin is involved in this, if you can imagine. But it then fuses into the membrane, so patch becomes part of the plasma membrane. It then moves up into that primary cilium, and it allows the glee to be released without being cleaved. So it then comes out full, and it becomes an inducer. So that's the signal that's coming from the ventral side, from the notochord, and from the ventral side of the neural tube. Neural tube. And that's it. That's all I got for today. It doesn't seem like much, but it doesn't seem like much, but it took me a long time to get that together. So any questions or comments based on that? Is this one of pathways in cell biology use? I still don't have a, nope. No good all, no all good. No all good, okay. When will our last exam be? It'll be during finals week, whenever we're scheduled for our final, it'll be at that point. Any other questions? Shall we call it a day here? How long will we have? Same as the last exam. It's just going to be an exam. It's not really a final. So an hour and 15 minutes should be good. It'll be very similar to the last one with like 6, not fill in the blank, but 6, choose the right answer, and then 4 short essay kind of things. Leo Lee, checking in. Aja. Cyan's here. Hello, Cyan. Justin. Okay, we're going to call it a day. Get some work done instead of this. Austin says, thanks. Let's call it a day. Thank you, Franklin. All right, I'm gonna stop it now. Thank you. So we're moving on to neural crest cells next time. How do I stop? I'm still recording. Stop. Stop video. Okay, video. Ooh, there's me. End the meeting.